Imagine this. Inside every single one of your cells is this um, incredibly detailed instruction manual, billions of letters long.
Dillions.
And here's where it gets wild there. This manual, it's been evolving and rewriting itself for, well, billions of years.
Hmm.
Okay, let's unpack this. We're diving into your genome, that amazing blueprint that makes you, you.
Yeah, and what's fascinating right from the start is thinking about our genome as this ongoing autobiography.
An autobiography.
Kind of, yeah.
Yeah.
Written not with pen and paper, but in the language of genetics, what Matt Ridley in our source calls genetish.
Yeah.
It's a record of our entire species' history, tracing back to the very beginnings of life on Earth.
Exactly. And this massive autobiography isn't just one enormous chapter, right?
Mm-hmm.
It's neatly organized into 23 pairs of chromosomes. 22 of these pairs are numbered, generally from largest to smallest chromosome 1 all the way down to 22.
Right.
Then you've got the sex chromosomes, X and Y, which determine biological sex.
And the number, it's interesting you mentioned that. Because those first 22, the numbering is more or less arbitrary, isn't it? It's not like genes that work together are necessarily grouped on the same chromosome.
No, it's not like a well-organized library where similar books are shelved together at all. It really highlights how our genome's organization is a product of evolutionary history, full of, twists and turns, rather than some perfectly engineered system.
That's a great way to put it. And our source for this deep dive, Matt Ridley's book, Genome, it beautifully shows how we can explore this vast landscape chromosome by chromosome, almost like turning the pages of this ancient story.
He does that thing like Primo Levi did with the periodic table, right?
Yeah.
Focusing on a key gene from each chapter, or chromosome in this case.
Precisely. He picks one gene per chromosome to sort of anchor the discussion for that section of the book. It's a really clever approach.
So your mission, should you choose to accept it, and well, you already have by listening, is to get a clear understanding of this incredible world without feeling like you're drowning in scientific jargon.
Yeah, think of this as your shortcut.
Exactly, your shortcut to the most captivating stories and insights hidden within your own DNA and the potential this knowledge unlocks. It's huge. Anthropology, psychology, medicine.
It promises to revolutionize so many fields. And this intricate story, it's built on a surprisingly simple foundation. Just 4 letters.
A, C, G, and T.
Oh. A, C, D. Those are the bases that make up DNA. And they link together in these 3-letter words, codons, which essentially code for the building blocks of proteins.
So picture DNA as these incredibly long chains, like strings of beads, with these A, C, G, and T beads attached. And usually it's not a single string, but that iconic double helix.
With twisted ladder structure.
Right, 2 strings wound around each other. Now, here's where it gets really interesting. This double helix has the ability to copy itself. It's replication.
Where A always pairs with T and G with C.
Exactly. And it can also be read, a process called translation, to create the proteins that do most of the work in our cells.
And that precise AT-GC pairing is just elegant, isn't it? That's the mechanism allowing life to perpetuate itself with such amazing accuracy.
It really is.
And while DNA holds the master instructions, there's another key player, RNA.
Right, RNA.
As our source highlights, RNA acts as a crucial intermediary. It's the messenger, basically, between DNA and protein production. But what's truly intriguing is the strong evidence suggesting that RNA might have actually come first.
Before DNA.
Potentially, yes. Serving as the ancestor to both DNA and proteins, a sort of primordial genetic system, the RNA world hypothesis.
It's just mind-boggling to think about these fundamental processes happening constantly right now inside every single cell in your body.
Absolutely.
And our understanding of all this, it wasn't always so clear. I mean, back in 1943, Oswald Avery conducted some truly groundbreaking experiments.
Avery's experiments, yeah, pivotal.
They finally identified DNA as the actual chemical basis of heredity. He showed you could change the genetic makeup of bacteria just by introducing purified DNA.
It seems obvious now, maybe, but back then.
It was revolutionary.
Yeah.
And not immediately embraced by everyone either.
No, it took time. Even Avery himself was quite cautious in his initial publications. The scientific community needed time to really grasp the implications.
While all that was happening, the weird versatility of RNA, its ability to copy itself, even catalyzed reactions that hinted at this earlier RNA world.
It really underscores the dynamic nature of science, doesn't it? How our understanding evolves as new evidence comes to light.
Okay, so let's dive into chromosome 2. Ridley titles this chapter Species,
Aptly named.
and starts with a rather surprising historical tidbit. Believe it or not, Until 1955, the accepted scientific consensus was that humans had 24 pairs of chromosomes.
24. It's amazing to think about now.
Right. It was just one of those things considered a well-established fact for decades.
And what's fascinating about that era is how long it persisted. It mostly stemmed from the work of Theophilus Painter back in 1921.
Painter.
He was looking at cells from tests, and despite some uncertainty in his own observations, he concluded 24 pairs. And subsequent researchers, maybe influenced by Painter's status or findings, kept reporting the same number.
Wow. So it just became dogma.
Pretty much. It's a powerful reminder that even in science, what seems like established truth can sometimes be wrong. It highlights the need for, skepticism and constantly reevaluating things.
Until 1955, when improved techniques finally nailed it down.
Exactly. 23 pairs. Confirmed.
Talk about a moment where science had to say, oops, our bad. But this sets the stage for a deeper look at what makes us, well, us as a distinct species. And Ridley takes us way back, billions of years, to the origins of life.
Introducing that concept of genes as survival machines.
Right, a term Richard Dawkins popularized. These genes have essentially been building bodies, these vehicles, for eons through relentless natural selection.
That idea of our bodies as survival machines, built by genes over vast timescales, it's a really useful way to think about evolution. Starting with incredibly simple, single-celled organisms, just focused on basic chemical efficiency.
And gradually branching out, getting more complex.
Through countless generations, only the most successful gene combinations, the ones that built the best survival machines, got passed on.
So when we zoom in on our own branch of this tree, the genetic similarities between humans and chimpanzees are, well, they're remarkable.
Around 98% similar.
98%. And get this, genetically speaking, we're actually closer to chimps than gorillas are.
That's often surprising to people.
And even if you look at the physical structure of the chromosomes themselves, 13 of them show no visible differences between humans and chimps.
That 98% similarity is just profound, isn't it? It really makes you think about how relatively few genetic differences, just that 2% or so, account for the significant ecological and social distinctions we see.
So what's in that 2%?
Well, the thinking is that even subtle changes, particularly in genes that regulate other genes, regulatory genes, and those involved in growth and development, can lead to really substantial differences in form and behavior.
So what triggered that split, that divergence from our shared chimp ancestors? There were a few ideas out there.
Like the East Side Story hypothesis.
Right. The idea that geographical separation may be caused by the Great Rift Valley forming in Africa around 5 million years ago.
Isolated our ancestors on the drier eastern side. A different environment, new evolutionary pressures.
Makes sense?
Yeah, the East Side Story offers a compelling explanation for those initial steps. The shift to a drier, more open grassland environment would definitely favor adaptations like walking upright.
Oh.
Bipedalism.
And we have evidence for that, don't we? The Laetoli footprints.
Oh, the Laetoli footprints in Tanzania, yeah. Roughly 3.6 million years old. Incredible fossil evidence of our ancestors walking upright.
And that transition to walking on two legs likely had knock-on effects, right? Freeing up hands for tools, maybe influencing social stuff too.
Definitely. Which leads to another fascinating hypothesis Ridley mentions.
Go on.
That the increasing trend towards monogamy in our evolutionary lineage might have actually helped drive the development of larger brains.
How so? Monogamy leading to bigger brains.
Well, the idea is that in a more monogamous system, both males and females face stronger pressures to choose mates carefully. This could potentially favor individuals who retain youthful features into adulthood neotenous traits.
Okay, youthful features.
And interestingly, these traits tend to be associated with larger, more developed brains. It's a potential link.
Huh. That's a really thought-provoking connection.
Yeah.
Mate selection based on looks indirectly driving brain evolution. It shows how different evolutionary forces can kind of intertwine.
Exactly. Social behaviors, mate preferences, they can have significant long-term biological consequences.
And ultimately, Ridley emphasizes that the fundamental differences that define us as a species, separate from chimps, are indeed encoded in our genes.
That relatively small 2% of the genome holds the instructions for our unique ecological niche. Our social structures, likely involving those regulatory genes, genes governing growth, hormone production, leading to the physical differences like, the shape of our feet.
Yeah.
Absolutely. Even people who argue strongly for the role of culture generally acknowledge that the foundational differences between species are ultimately genetic.
There's that thought experiment, right? If you put a chimp nucleus in a human egg,
Right. The resulting organism, despite developing in a human environment, would still be a chimpanzee. It really underscores the fundamental role of the genome.
Now, within this chapter on species, Ridley also takes a moment to bring in some foundational concepts in genetics. He talks about Archibald Garrod.
Garrod, yes. And alkaptonuria back in 1901.
Garrod noticed this rare metabolic disorder ran in families, especially where cousins married.
Recessive inheritance.
And he proposed this groundbreaking idea of inborn errors of metabolism, the hypothesis that a single gene is responsible for producing a single enzyme.
It was truly revolutionary for its time, connecting a specific genetic defect to a specific biochemical problem. A major turning point.
And he correctly identified it as recessive. You need 2 copies of the faulty gene.
But what's remarkable is that his core insight genes make proteins, essentially, since enzymes are proteins, wasn't fully appreciated or integrated into mainstream genetics for decades.
Decades. And speaking of foundational figures, we get Gregor Mendel.
Ah, Mendel and his peas, mid-19th century.
This Austrian monk just meticulously crossbreeding pea plants in his monastery garden.
Uncovered the fundamental principles of inheritance. Particulate inheritance traits passed down as discrete units, genes not blending together.
His systematic quantitative approach, it laid the groundwork for modern genetics.
Absolutely monumental. But like Garrod, his work was largely ignored at first.
Until the early 20th century when it was rediscovered. Amazing how that happens.
It really is. Shows how important discoveries can sometimes take a while to sink in. And then there was the whole reconciliation of Mendelism with Darwinism, thanks to people like Ronald Fisher.
Right. And to round out this early history, Ridley touches on Hermann Muller's work in the 1920s.
Muller and inducing mutations.
Yeah, showing the genes could actually change, providing the variation that Darwinian evolution needs to work.
That was pivotal. It connected Darwin's theory, which relies on variation, with Mendelian genetics, which initially seemed more about stability. Muller showed how you get that variation.
And Ridley also loops back to Friedrich Miescher.
The guy who first discovered DNA way back in 1869.
And Miescher apparently had this surprisingly insightful suggestion about its potential role in heredity.
Even though at the time everyone thought DNA was just too simple a molecule to carry complex biological information, they favored proteins initially.
Shows how sometimes the earliest intuitions can be spot on, even if the proof takes a while.
Exactly. Early insights can be profound, even if their significance isn't immediately obvious.
All right, let's move on. Chromosome 4, which Ridley titles Fate.
Fate. Evocative.
And he starts this chapter with a really important critique, I think. He criticizes how genome catalogs often define genes primarily by the diseases caused when they malfunction.
Right. Using that analogy, it's like defining a liver only by cirrhosis or the heart only by heart attacks.
It's a really insightful way to put it. Focusing only on the disease gives such a limited, sort of negative view.
For so many genes, all we currently know is what goes wrong. It tells us nothing about their normal, healthy function. And it can create this misleading impression that genes are mostly there to cause trouble.
Which they're obviously not.
No. But Ridley does use Huntington's disease as a key example here.
The first known, completely dominant human genetic disease.
Exactly. One copy of the mutated gene is enough to cause the condition. He mentions the late onset, the potential for increased severity when inherited paternally.
And Nancy Wexler's search for the gene.
Her incredibly determined, decades-long search working with those large families in Venezuela. A remarkable story of scientific dedication leading to the identification of that specific mutation.
It's a powerful example of a single gene having a very strong, almost deterministic effect. A clear link between gene and fate, in a way.
A very clear link. But Ridley quickly pivots from that.
To emphasize that most traits aren't like Huntington's at all.
Exactly. Things like our facial features, most characteristics, they aren't determined by single genes, it's complex. Interactions between many genes plus unique variations from our parents, it's often quite, as he puts it, messy.
Polygenic, influenced by lots of genes.
Oh, right, polygenic, and often interacting with the environment too, which makes pinpointing the genetic basis for most traits much, much harder.
Which leads us into his discussion about asthma.
Ah, yes, asthma. A condition that's become much more common.
And Ridley really challenges the simple idea that this rise is just because of an increase in asthma genes.
He explores various environmental theories instead. Increased urbanization, maybe? Higher living standards? The hygiene hypothesis?
That idea that being too clean, less exposure to microbes early on, might make our immune systems overreact later.
Exactly. The core point being, Our genes haven't changed that dramatically in just a few decades. Something else must be driving that rise in asthma prevalence.
It really highlights that complex interplay between genetic predisposition and the environment.
Absolutely. There are genetic factors making some people more susceptible, sure, but the huge increase points strongly towards environmental triggers. And the difficulty scientists have had in finding specific asthma genes that hold up across different populations, that underlines the complexity, too.
Might be different genes involved in different groups even.
Could be. It's complicated.
Which naturally brings us to that long-standing, often really heated debate about the heritability of intelligence.
Nature versus nurture. The classic.
Ridley acknowledges the obvious huge influences of environment, upbringing, learning, culture, which has led to a lot of skepticism about purely genetic explanations for intelligence.
And understandably so. It's clearly not just genes. The debate is about the relative contributions, the how much and how.
It's one of the most complex and, frankly, often controversial topics.
Definitely. Both genes and environment play crucial roles. The challenge is understanding how they interact and the extent of each contribution.
He then discusses Robert Plumman's research.
Plumman's study on highly gifted teenagers, yes, finding that slightly different sequence in the IGF2R gene on chromosome 6.
Suggesting a possible genetic link, however small, to intelligence.
Yeah, but it's important to frame that correctly. Plumman's study, while interesting, really shows how hard it is to find specific genes with a big impact on complex traits like intelligence.
Right. Even if IGF2R plays a role, it's likely one of many, many genes.
Each with a small effect, interacting with each other, interacting with the environment. Finding a correlation isn't proof of direct causation either.
Ridley also, importantly, touches on the dark history here.
Yes, the historical misuse of intelligence testing. Figures like H. Goddard, the eugenics movement.
Using biased, culturally insensitive tests to draw really prejudiced conclusions about innate intelligence differences between groups. It's a cautionary tale.
A deeply disturbing one. It highlights the dangers of oversimplifying genetics and ignoring environmental and cultural factors.
He then shifts to explaining how scientists try to estimate heritability twin studies.
Comparing identical and fraternal twins, especially those raised together versus apart.
And adoption studies.
Right. Looking at adopted children compared to their biological and adoptive parents, these studies generally suggest genes do play a substantial role in the variation of IQ within a population.
With estimates often landing around 50% heritability.
Yes, but it's crucial to understand what that 50% means. It reflects the proportion of variation in IQ within that specific population studied that can be attributed to genetic differences within that population.
It doesn't mean 50% of your individual intelligence is fixed by your genes.
Exactly. Nor does it mean intelligence is unchangeable. Heritability is a population statistic, not an individual destiny.
Ridley gives a bit more detail on that IGF2R gene. Mentions its connection to insulin-like growth factor receptors, glucose metabolism.
And even a study suggesting higher IQ individuals might use brain glucose more efficiently during learning tasks. A possible biological pathway, though still tentative.
And then there's that really surprising correlation he brings up.
Between IQ and body symmetry.
Yeah. Things like having ears or limbs that are very similar in size on both sides. Developmental symmetry.
Which is thought to be a marker of developmental stability. Less fluctuating asymmetry might mean less developmental stress during growth. Fewer infections, better nutrition, fewer toxins encountered, perhaps.
So the idea is maybe the heritability of IQ is partly reflecting an inherited resistance to those developmental stresses.
It's a fascinating idea. It suggests that genetic factors influencing our general robustness, our ability to develop smoothly despite environmental challenges, could indirectly impact cognitive development. It really blurs those simple lines between nature and nurture.
It makes it much more intertwined.
Definitely.
He also wraps up this section by mentioning James Flynn.
The Flynn effect.
Huh.
The observation that IQ scores have been steadily rising for decades across many populations.
Which can't be due to rapid genetic changes.
No, genomes don't change that fast. Which leads to Ulrich Neisser's theory.
That our modern environment, especially the visual environment, ads, films, complex graphics, video games, maybe actually enhances some of the abstract problem-solving skills that IQ tests measure.
It's a compelling argument for the powerful role of the environment. And it further underscores that dynamic interplay between genes and the world around us.
Ridley even touches on the idea that our genes can influence the environments we seek out,
Right. The genetics of nurture. If you have a genetic predisposition towards, say, reading, you're more likely to spend time in environments rich with books.
which then further develops those skills. Your genes shape your environment, which shapes you. It's a feedback loop.
Exactly. Like the heritability of short-sightedness being partly due to the heritability of reading habits, it really complicates that simple nature versus nurture dichotomy. It's nature via nurture, often.
So let's try and quickly recap some key takeaways from this deep dive today. We've journeyed through the human genome as this um, incredible, billions of years old autobiography.
Written in Genedish.
Right. We explored our kinds of evolutionary ties, especially with chimps, and how just a small percentage of genetic difference can lead to major distinctions.
That 2% doing a lot of heavy lifting.
We've also delved into the complexity of how genes influence traits, moving way beyond one gene, one trait thinking, especially for things like intelligence.
Highlighting that crucial constant interaction between our genetic inheritance and the environment we experience, create, and navigate.
This exploration of just a couple of chromosomes, based on Ridley's work, really shows the immense scope and complexity.
It reminds us that while our genes provide a kind of framework, our environment, our experiences, our choices, they all play vital roles in shaping who we actually become.
And what's truly fascinating is just how much more there is still to discover within this incredible inner autobiography.
Absolutely, we've only scratched the surface.
And on that note, here's maybe a final thought for you to consider. As we keep unraveling the secrets within our genome, what are the profound implications for how we understand ourselves, our health, our potential, our place in the story of life?
How might this growing knowledge continue to reshape our understanding of our origins, maybe even the definition of what it means to be human?
Our inner story is definitely still being written. And the next chapters, well, they promise to be full of even more surprising revelations.